Okay, this is the second prep for lecture 2, and they won't all be this long, but if you just bear with me, oops, um, I just want to make sure we get through sort of the basic material, especially at the beginning of the course when we're just trying to make sure everyone's got the same kind of basic level of understanding. So, how can we actually tell chromosomes apart? This should be an actual question. Oh no, not a question. Um, so when you look at something like this diagram, you can see differences between the chromosomes. So this is chromosome 1, the pair. They're a certain size and they're a certain shape. They have a certain position of the centromere, and so on. And so you can tell the individual chromosomes apart based on those sorts of things. We can use um, the size, we can use the shape, and then sometimes there's ways of staining the chromosomes that shows different kind of patterns of bands on the chromosome where the stain is really dark and really light. And each different chromosome has its own unique pattern. And of course, when we're looking at a particular species, we use this information, the size, shape, and those features to distinguish the different chromosomes within that species, our chromosome 1 compared to our chromosome 5 and our chromosome 10 and our X chromosome and so on. But when we look between species, we're going to see differences as well. They're not all going to have a karyotype that looks the same. Even pretty closely related species will often have some differences. And they'll have especially differences in just the number of chromosomes, right? The size can vary quite dramatically. If you look back here, this is a huge chromosome that probably has thousands of genes on it, and some of these tiny ones might have 100, or around that. So there might be huge differences in sizes that we can observe within one particular organism. Okay. And then the chromosome number, we've already kind of gone through this, so I won't go through it again, except to say that for humans, n equals 23 chromosomes, 2 of each, so 2n equals 46. And if any of these things are unclear, just email or ask me or one of the TAs after the lecture, and we can talk you through it. So when we talk about chromosome shape, what are we talking about? Because I mentioned shape, but I didn't say exactly what we meant. And so each chromosome has a centromere. A centromere is, if you look at the chromosomes, they often look like a constriction. So if we look here, you can see there's a narrowing here, and you can see that it looks like there's just one strand of DNA here, whereas there's 2 chromatids and so on. And so it's like a narrowing in the chromosome that is where the centromere. This is where the spindle will attach during cell division to separate either chromosomes or chromatids, depending on what exact division it is. And when we're doing shorthand, we usually draw the chromosome like a stick, and then we have usually a circle that indicates the position of the centromere. So it doesn't physically look like what the chromosome would look like under a microscope, but it's the way we represent it. Telocentric means the centromere is at one end. And this is an example. All these ones are telocentric because the centromere is right at the end, and there's just only one set of arms. Acrocentric, there aren't really acrocentric chromosomes. Well, a little bit. So chromosome like this, where the centromere is not in the center, but it's clearly off to one side. So you've got one much shorter and one much longer arm. That's acrocentric. And then most of these two anyway, I would call metacentric. Meta means middle, and that means the centromere is in the middle, and one arm and the other arm are approximately the same in size. Okay, so only one arm showing here. 2 arms are showing. One is quite a bit shorter than the other, and then here 2 arms again but they're approximately equal in size. Um, so yeah, and there is another category, but I don't think it's really necessary to go through that. Those are the three that we'll talk about. So acrocentric is off center, short arm and long arm. Metacentric is the middle or close to the middle. If this was just slightly off to one side, we'd still call it metacentric because it's pretty much close to the middle. Okay, so let's move to the arrangement of genes on the chromosomes. If we look at single-celled organisms like bacteria, which are prokaryotes, and yeast, which are eukaryotes, we can see this is a kind of a pattern. You have 20 kb means about 20,000 nucleotides in length. We'll talk about what that exactly means when we talk about DNA structure, but that's an amount of DNA, and there's lots of genes. 1, 2, 3, 4, 5, 6, 7, 8. So 13 genes in this short region of 20 kb. So the genes themselves are pretty short, and there is little narrow spacers between them. And we're going to talk a lot about the kind of DNA sequence that's between the genes and what's the regulatory versus the coding sequence and all that sort of stuff. We'll cover in more detail later. But when you look at both of these types of organisms, you can see the dashes here just mean that there's more DNA to either side of this is just one little piece of a chromosome, but you can see that the genes are really close to each other, and they tend to be relatively small. When we look at what we would call higher eukaryotes, like fruit flies and humans, here just notice this is 200,000, so this is 10 times as much DNA as this, and we have 1, 2, 3, what is it, about 7, 4, 5, 6, 7, so 8 genes, right? So 8 genes compared to 13, but there's 10 times more DNA here, so the genes are bigger. And they're also, there are spacers. So if you look at here, if you think about a gene encoding information, protein information, the information that's going to make the protein is the dark green. And then there's little spacers in between those that doesn't end up forming part of the protein. We'll talk about regulation and sort of splicing and things like that later. But just to know that the genes are more complicated. They have spacers and so on. There's no spacers in these genes. And there's also large regions in between because the regulation of a gene, when it's turned on and off and what kind of cells and so on, is more complicated in higher eukaryotes. And so there has to be more sequences that determine, whether this gene is being expressed or whether it's being shut off and so on. So there's a number of things to notice compared between the prokaryote and the very simple single-celled eukaryote and higher eukaryotes. Okay, so we look at chromosome structure. A human cell contains about 2 meters of DNA, so that's 1 meter per genome inside the nucleus. But the nucleus is really tiny, 0.006 millimeters, so that is 6 microns, which is 6 millionths of a meter. So it's a really, really much smaller amount. But of course, the DNA is very narrow. It's 2 nanometers wide. So how do you get this really long, 6.5 feet of DNA into a nucleus that you can't see with your naked eye? And you do it by folding the DNA up in a very, very, very compact structure. So the DNA is wrapped around nucleosomes. The DNA itself is 2 nanometers wide, approximately. And then when it's wound around nucleosomes, which are special octamers, octamer means eight, so there's eight of these little proteins called histones that form the kind of bead, and then the DNA wraps around it, and that compacts it about 5 times. So it's shorter, but it's now 5 times wider. And then and this is called beads on a string, because I'll show you why in a minute. And so then that strand is coiled around. So you've got the beads on a string, which is the DNA wrapped around the proteins, and then that is coiled around, kind of like a spring. Oops, but it's done more neatly than this. And that makes it about 3 times more compact. So it's shortening again, but it's widening by 3 times. And then those loops, they loop around a scaffold, which is in some other proteins that we're not going to talk about. And this forms approximately 300 nanometers, so 10 times more compacted. And they form interphase chromatin. So this is the chromosomes when the DNA, or when the cell is not dividing. It's just sitting around doing its job, being a cell. It's in between the division, the cell division. In preparation to actually divide, the chromosomes get compacted even more, and the 300 nanometers are folded over even more to make around 1500 nanometers in width, for maximal compaction. This just makes the process of dividing the chromosomes into 2 new cells during cell division much easier. And when we talk about chromosomes and we draw them, we draw them this way. You've seen them drawn like sticks with the dot. We always draw them as if they're compacted, because it's ridiculous to try and draw them when they're all unwound, and so on. This is a really good picture that kind of shows that. This is an interphase human nucleus. It's been probed, hybridized with little probes that detect certain kinds of sequence, and then the computer has false colored it, and it colors it so each chromosome is recognized by a slightly different color. So there's I think this purple here and this purple are actually the same. And so this is one copy of this chromosome, and this is the other copy of it. Now this is a rounded structure, so the rest of this chromosome is kind of around behind where you can't see it very well. And here's a chromosome that's sort of in behind another, this bright blue one, and then here's that chromosome kind of spread out somewhere else. And so this is how they're arranged when the cell isn't dividing. Here's a dark blue part, and then here's the other copy of that chromosome over here, and so on. It's a really beautiful image, though. But during metaphase, instead of occupying these, so this is where the 300 nanometer chromatin is found, and then it's much more compacted, about 5 times more compacted, to make these metaphase chromosomes. These ones have been stained in a similar way, except that each chromosome has a unique sort of color pattern. This is computer generated based on just intensity of staining and so on. But it's also a beautiful picture. And I'll just point out, I've already mentioned that human chromosomes are numbered from largest to smallest, with the sex chromosomes always shown at the end. But I also just point out that it's really hard to see the sister chromatids in these chromosomes. These are getting ready to divide, so they have replicated, but they don't look like we usually draw them with the easily seen separate sister chromatids. But they're there, and if you look really carefully on some of these, you can kind of see, like on chromosome 16 for this one, you can kind of see there's a little dip on the top and the bottom. The chromatids they're close together, and so it's easy to forget that these are actually replicated chromosomes. One of the reasons why I showed the rat one is it was treated so that you can see the sister chromatids really, really well. This one isn't too bad either. You can see that there's two sister chromatids, at least on this side. Um, yeah, and some of them you can see here and here and so on. So that's just something to bear in mind, because when you see the pictures like this, I don't want you to forget that these are replicated chromosomes, and that the right way to represent them in drawing would be like this to show that they have replicated. They've now got two copies of the same chromosome attached to the centromere. So the total length of the amount of compaction is really quite interesting if you think about the fact that um, the level of compaction is really huge. And if you took the number of cells, the approximate number of cells in your body, because there's a number of different estimates that vary somewhat, but according to NOVA, if you took all the DNA strands in your body and matched them end to end, they would go 6,000 times as far as the distance from the moon to the Earth, which is really quite astonishing. So, and I've seen smaller and larger estimates based on the exact number of cells that are thought to be in the human body. It's going to vary, of course, with individuals, but no matter, it's a really large number, is the point. And this just shows a little bit what the kinds of compaction I was talking about. So you've got, your DNA by itself, 2 nanometers wide. When you wind it around the histones, that's what these little cylinders are, then you widen it to be 10 nanometers wide. And then when these coil around in what's called a solenoid, or a spring structure, then they widen to 30 nanometers and get shorter. And then when this coils around on itself, again, it makes a wider structure. And then when it's looped around on scaffold, it gets even wider. And so this just shows you the huge amount of folding and so on that goes on in cells. And in interphase, most cells that are not dividing, this is the state of the chromatin, but when it's getting ready to divide, it gets even more compacted. So how does information become an actual form, a phenotype, a physiology, a functioning enzyme, or what have you? So the DNA is transcribed, and it's transcribed to produce RNA. And we will be spending entire lectures on these processes, so this is just introductory. So the messenger RNA is made. In eukaryotes, it goes out of the nucleus into the cytoplasm, and then it gets translated to produce protein. And in the case of hemoglobin, it produces a protein that is found in red blood cells and is responsible for binding oxygen molecules and carrying the oxygen around your body. If you're taking 205 or have taken 205, you'll learn more about that. You've probably learned about it to some extent already in other courses. So if you have normal copies of both of the genes, you get the normal hemoglobin protein. The molecule folds up properly. There's 4 of them in your red blood cells, and they cause the red blood cells to have this funny sort of dish shape. They've got a narrower end. And these ones, as it happens, don't have a nucleus. So this is one of the few cell types in your body that doesn't actually have a nucleus with all the genes. So if you have 2 normal copies, your blood cells are happy. They are carrying around oxygen very well, and you're very healthy in that regard. However, if there are 2 defective copies of the gene, then that means all of the hemoglobin protein is defective. And we're going to go through the reason for this later in more detail in another section where I talk more about mutations. But the protein that doesn't fold right, and what it does is it deforms the blood cells so that they have this funny sort of sickle crescent kind of shape. And because they don't carry oxygen very effectively, people are very, very sick. They have a ton of pain. They're always short of, low levels of oxygen and so on. And so they're very sick. It's an awful condition. If you have one good and one bad copy of this gene, you have some normal functioning red blood cells. They are able to carry oxygen, and so you're usually okay. But if you're at high altitudes, it's in condition, you may remember that if you are at a high altitude, the air is thinner, there is less oxygen available, and so you can suffer from severe altitude sickness and have real problems in any kind of low oxygen situation. So this is just a quick example, and it's introducing something I'll be talking about later in the class. Almost everything I talk about here now, we will be talking about later in the class. So the two different forms of the gene that I just talked about are called alleles. So everybody has two copies of the hemoglobin gene, but they may differ in DNA sequence, and it may be that both copies give a functional protein. It could be that both copies are defective in some way. They have a mutation. It's not producing a functional or very functional protein, and so you have this condition, sickle cell anemia. And most genes, I'll just point this out, they have more than two alleles. A lot of genes, there are maybe, half a dozen or could have more different variant alleles. But most of the examples we're going to look at, especially towards the beginning of the class, we're only going to look at two alleles. It turns out for this particular mutant allele, the DNA sequence is different from the normal gene by just a single nucleotide, which means that the RNA that gets made via transcription, has a single nucleotide change as well. And then the protein that is made by translation, excuse me about that, has one amino acid that's different. And that one changed amino acid causes protein that folds so badly that it causes this terrible sickling and this horrible disease. There's a huge amount of pain, as well as difficulties with, just no energy, because you're really not getting enough oxygen to your tissues at all, and other things as well. You can look up sickle cell anemia if you like. Those of you who are interested in medicine will probably find it of great interest. Okay, so this brings us to two very important terms, genotype. A person's genotype, or a plant or a fly's genotype, is the alleles that are present. So they could, in the case of the example, they could have two good copies of the hemoglobin gene. They have two of the same normal alleles. They could have one good and one mutant one, so they have two different alleles. Or they could have two same alleles that are both the mutant form, right? So that would be their genotype, and we would have a way of writing it, H indicating the wild type and little h indicating the mutant, and then you could have a genotype that is listed this way, or this way. And we'll talk about writing a genotype later, but this is the idea. It's a way of designating what actual alleles the individual has. But the phenotype is how is that expressed? Because sometimes you could be like this with a good copy and a bad copy of a gene, and your behavior, your physical appearance, your physiology, your health, everything is perfectly normal. Right? So the phenotype will be the appearance or the physiological expression of that genotype. And we'll look at, when we talk about Mendelian inheritance and look at dominant and recessive alleles and so on, you'll see more about what that means. So the phenotype of an individual will be variable. Each one of us, our DNA is remarkably similar, all of us, me and the TAs and you in the class. But we all have some differences, and that's due to the allelic, the genotypic differences at multiple genes. And as I said, some alleles you can't necessarily observe depending on what other alleles the individual has. and what environmental factors there are, and so on. So, for example, an individual who has one mutant copy and one good copy of the hemoglobin gene will be normal. And if you were to just look at them, you wouldn't know that they had, that one mutant copy unless they were under low oxygen conditions, and then the vulnerability shows, right? So this shows you how you might not have an observable phenotype based on your genotype, but there might be environmental factors that affect whether you see a phenotype or not. Hopefully that makes sense. And then when we're talking about genetic variation, there's a huge amount of variability, of course. I mean, despite the fact that humans have very low genetic diversity, we're very, very similar to one another. We do have, of course, polymorphisms. Just think of all the different hair color shades there are in the world, or eye colors, and so on. There's lots of variation. And so any kind of variation like this is called a polymorphism. Poly means many and morph means form, right? So there's polymorphism based on average height, based on eye and hair color, skin color, all kinds of things like that, where there's just a huge amount of variability. The wild type is a term that generally refers to the functional gene that works normally in wild populations. So, for example, you look at zebras; they're all striped, and they have black and white stripes, and they have a certain general body size, and so on. And so, in most organisms, when we talk about wild type, it refers to the most common form of the gene, and usually, when I'm thinking about it, I'm not usually thinking about hair color, for example, because while it's not abnormal to have lighter hair, for example, in humans, right? It might be abnormal in a wild animal, but it certainly doesn't make any difference in humans. And so when I think about wild type, when I'm thinking human, I don't usually think sort of things like eye color or hair color. I think about, say, a liver enzyme, right? If you have a functional liver enzyme that catalyzes some reaction in the liver, then that's the wild type. And the mutant form is generally if there's a variant form of the gene that doesn't function normally or not as well as the wild type. And you could have, for that particular liver enzyme, there could be multiple variant forms that are just a little bit not as efficient or effective as the wild type form. But these are terms that are used a lot, and more so, we use these kinds of terms with generally not really with human populations. So that's the only kind of human example I can think of. But if we're talking about fruit flies or lions on the Serengeti or something, these are terms that we would normally use. Okay, so that concludes the introduction of basic terms. If you've chosen to, I will be putting, I haven't done it yet, but there will be a couple of textbooks available to read up. Some students really like to have that. I'll make them available in the library so that there's no cost to you, but you can look at them if you want to. And yeah, I'm just trying to remember what my point was. Okay, I'll just leave that for now. So just this review hopefully introduced. Oh, I was going to just say that there's other material in the intro chapters for all the textbooks, but I tried to pick out for these lectures the ones that are most relevant and are going to prepare you best for the material we're going to be covering through the semester. Some of the material will prepare you, which I haven't included, prepare you for upper division additional courses that you can learn if you're interested and want to take those courses. On the next lecture, I'm going to do a two-stage review exercise where you will do some individual just answering of questions, and then you'll work together in groups in the lecture period to answer those questions. And it's going to help just point out to you sort of basic background that we want you to have before you proceed through the course. that's going to make life easier for you if you have that basic information. Um, and then we'll just launch in. So the next topic is going to be DNA structure and replication. I will have some prep again, and then we'll go through the steps of DNA replication in the lecture. One thing you should do is, as you prepare, as you listen to these recordings or read materials or go through some of I'll probably have some little shorter videos and things to introduce topics. You should try to make a note of any questions you have as you prepare. If something occurs to you, make a note of it. Stop the video or stop reading for a moment and write it down so that you're really engaged as you progress through. It's useful to make your own glossary of terms that you write down and explain in your own words what they mean with little diagrams or flow diagrams or any kind of way you can think of that will help make it easy for you to understand and explain what those terms mean. Not a bad idea to find a set of other students who have the same aims you do. If you want an A plus or nothing, and you don't want to necessarily pair up with students who aren't that interested in genetics and just want to get through, right? But if you just want to get through and the rest of the group just wants to get through too, you can form a group and you can help each other with the material, quiz each other, make questions, and discuss topics, make notes of the stuff you found difficult and see if the other people in your study group can explain it, and so on. There's going to be opportunities for practice weekly on the Code on site, and I haven't finalized that yet. But when I do, by the time you are listening to this, that will be finalized and I'll have it available. Yes, and I think that's basically it for now. See you in class.